On this episode of Translating Proteomics, host Parag Mallick speaks with special guests, Doctors Ben Neely and Ben Orsburn, leaders in the proteomics community and hosts of the Proteomics Show podcast. Their insightful conversation focuses on the role of communication in proteomics and covers impactful things they've learned while hosting the Proteomics Show, how their communication efforts have changed their research, and barriers to proteomics becoming more popular in the life sciences and in the broader public. Now here is your host, Parag Mallick.
All right. Welcome back to Translating Proteomics. On this episode, we are really fortunate to host Ben Neely and Ben Orsburn ahead of US HUPO 2025. And Ben Neely is a research chemist at the National Institute of Standards and Technology in Charleston, South Carolina. At NIST, he focuses on the development of new materials for omics applications and the generation of standardized data across non-model species. He additionally works to develop methods for emerging proteomics applications, does a lot of work in data-independent acquisition and metaproteomic analysis. He got his PhD and postdoc from the Medical University of South Carolina, where he focused on environmental microbiology. We're also joined by Ben Orsburn, an assistant professor at the University of Pittsburgh, where he uses and develops single-cell proteomics techniques to study human biology and pharmacology. He did his graduate work at Virginia Tech and his postdoc at Hopkins. He held a variety of industry positions and was a principal investigator at Johns Hopkins. And throughout his career, Ben has leveraged his knowledge of mass spectrometry, proteomics, and protein informatics to advance biology and medicine. He also runs a hugely popular blog called News in Proteomics Research, where he covers the latest developments in proteomics. And I would be absolutely remiss in mentioning, if I didn't mention, that the Bens are the co-hosts of The Proteomics Show, a podcast sponsored by the Human Proteomics Organization, and to my knowledge, the very first proteomics podcast in the world. It is an exceptional podcast, and we're really delighted to do a crossover today. They interview proteomics researchers in a fireside chat format, and their conversations cover not just research, but everything about their guests, their hobbies, their motivations, their histories, and you should definitely check it out. It is fantastic. And so both of you, welcome so much to Translating Proteomics. I'm so delighted that you're here today.
Yeah, thanks for having us.
And thanks for the intro. I've made me feel really smart there for a second. It's probably just that I didn't have lunch.
It is entirely accurate in every way. I'd love to dive in today just to chat about the Proteomics Show and how it came about and what your goals are for it. And I'll turn it over to whichever of you wants to take that first.
I feel like that's you, Osborne.
Yeah. Okay. So I ended up having a really crappy commute for a while, and the drive into Baltimore isn't always the most fun thing in the world. And there's a stretch of road that's actually called the Thunderdome because there for a nice stretch of time, one person would not leave alive every day. So this nice 45 minutes to an hour and a half drive, I just thought it would be really fun to have something to listen to. Ben and I were sitting outside US HUPO Charleston, in the middle of the day, and then we're just like, okay, we're just going to do it. I think that was the conversation. Like nobody else is going to do, so we're just going to start a podcast and see what happens.
We
had
wanted
no idea
other
to make a
people to
podcast.
do it.
What?
Yeah, the goal is we wanted it, but no one did it. Right? Yeah.
So did you try to encourage specific people and say, hey, you should do a podcast, and they just didn't pick up the mantle? Or you were just thinking it in your head, there really needs to exist a podcast about proteomics?
We just thought it. Strong thoughts.
I mean, I don't listen to podcasts. It's not a medium but I was like, okay, well, I really try to read a paper every single day. And it's like, man, could I just get around that and listen to a paper, right? Can I learn something about proteomics and then not read a paper today, right?
No, that's amazing. And I guess just taking that one step further, first off, what are the goals of the podcast? What are you trying to do? What do you want to get out of the podcast?
Uh, Goals. Well, I'll give you my goal. My goal is, and I think this actually goes back. The other part of that conversation was like, what if we had this show and it gave us an excuse to talk to like cool people? Like that was, and I think like the person actually Ben mentioned in Charleston was actually someone we had as a guest, and I think that was the goal. I mean, it's, we've given, I think we've talked about this some, but the doing something for yourself is kind of an easy way to do it. So, I mean, I think there may be some other goals that, Ben, like we had a season where there were some goals, but I think mostly to me, I think it's pretty selfish. Like it gives me an excuse to talk to people that are very interesting.
Yeah.
And like people seem to not hate it. So,
I think it's the exact opposite. I think people love it,
yeah.
and they are religious followers of it to the point that you did a live podcast at US Hoopo in the past, and it was incredibly well attended.
Yeah,
That
I wasn't there. I was sick, but but that is my favorite one because I didn't have to listen to my own voice.
and, but
And,
even
and
but even
Robinson
like
killed.
yeah, but
She's
even like that one, like we didn't want to do that. Like so Ben was supposed to be there. But they were like, oh, we really want you to do a live podcast. We're like, why? Who is going to show up and listen to us? Because even if you go back, like the first, so we started talking about like in Charleston, and the next year we were at US Hupa and we had done like the episodes, and it was like we knew people listened, but it was weird to have all these people come up. Do you remember in Chicago? And they're like, "Oh, I love your show." I'm like, kind of look at them weird. And then in Portland, they wanted us to do a live show, and we did it. And yeah, you're right. Like this room was packed, and Osborne, you weren't there, but they were just like yeah, it's weird for the engagement because I for one don't think about people like who's on the other end. Like I try not to think about it. Um, but yeah,
Well,
yeah.
you sort of it definitely has turned you into proteomic celebrities, which is sort of a strange concept that there exist proteomic celebrities.
That's
But
funny.
but if in the pantheon of proteomic celebrities, I think your podcast has helped bring proteomics not just to other proteomics people, but to the wider community. So we'll come back to that later. But I'd love to also, you mentioned having selfish interests and wanting to chat with people, but there also, I guess there's a little bit, Ben, you mentioned it would be fun to be able to listen to and talk about papers. And so are there other aspects scientifically and with regards to your research where there's been some interplay or between the podcast and your own research?
I have an example right now that what we were interviewing this. So we did a little sub season for Hoopo's early career researchers, and so they were all undergrads, right? I guess one of them had just graduated, so he's doing a semester. But they were, so these young students, and one was telling us about how they'd flow sorted, all the different cell types in a mouse brain, and was doing proteomics on them. And we actually randomly sorted cells from mouse brains, from wild type of CRISPR knockouts, but we're having trouble making sense of the data.
Mhm.
And so JC Price just sent me like 2 days ago unpublished data that is like the rank abundance of all of the proteins from every cell type in the brain. And we're absolutely going to collaborate on this because now my paper makes sense, right? Well, it's Colton's paper, but and I actually have something to show at US Hoopo because I was planning to show this. And it's just like, here's some gobbledygook and there's a big, PCA of 4,000 cells, but I don't know what any of them are. So yes, so I've got a good example from right now.
That's
I
amazing.
from that same show this morning, I went to my colleague and presented to him the whole C half. So this is Chad Heyer, and he was talking about the iodine and the C half labeling and protein stability. So yeah, again, like I think we lean into our show. Like we don't pick them normally. We've picked a few, but they just kind of they're it's the result of other organizing and picking and prioritizing. But for I think like Ben just said, we're definitely heavily influenced by these people that we're talking to. Um, so for better or worse, I guess. Um,
Right.
Well,
And Ben kind of alluded to this, but we did have a targeted season where we went in with goals, where it was like, okay, the test, like, could we use this as an outreach tool? And so we did this little, what is it, 6 episodes or something, where each one was, what is a protein? Why do proteomics? What proteomics can't do? And I think that we'll look at the stats at
I
some
assume that
point.
was a really short episode.
Oh, oh no. So it was six different episodes, I guess,
No, I meant
right?
the specific one on what proteomics can't do.
It
It was
Oh.
it's actually ridiculous because we started off just talking about like it can change the oil in your car. Like it can do everything. Like
Right.
it's completely did it. It failed at that question.
Yeah. The response from a stats perspective is really like the listenership is really good, but it didn't hurt having Matthias Mann as one of the guests, right? So I don't know where the numbers were before, but it definitely looked like we got a boost there.
that's
Um,
that's really interesting. And so, if you think about over the course of these many, many episodes, what is the most surprising thing you've learned from your guests, either about your guests or about science itself?
Nobody will believe me when I say this, but I think that every scientist is actually really interesting. And sometimes you have to dig a little bit, but everybody's got like like something outside of the lab that they're passionate about. And that's really fun to get to, right? Um,
And they all will start off without fail and say, I am boring. This is gonna suck. And then we
yeah.
will talk and they'll have a blast. I mean, I think maybe I can't think of anyone that hasn't said like, oh, this was not fun. And each one, I don't know what the what the formula is, but sometimes we definitely unravel a little more. Whether it's Ralph Bradshaw talking about ghost cities or John Price, me trying to talk to him about how he went to the high school where Napoleon Dynamite was filmed, like whatever it is, like Ben saying, like every person has this fun thing. And so what I love, and if you go way back to the show, like, and you can kind of see this as Ben and I have developed over whatever 70 episodes, is you Osborne tends to be he's like the technical like more technical things, I feel like. And he will go deep. Well, okay, whatever, but you'll go
Okay,
deep.
whatever.
And
Sorry.
I will like tug on these other strings that might be like, why did Chad Hire build a thing, a t-shirt cannon? But then like we're both going into both of those areas. But I think that was like the original priority. Like we wanted to be really technical. I was more interested in the personal side, but now it's this really fun blend.
Yeah.
But yeah, I completely agree with Ben. People are always interesting and they're always convinced they're not. And except for you, Pruk, like you like know that I feel like you're like, yes, I have this like amazing, like there's some people that you're like, okay, sure, this is obvious. But most people, they just think they're lame and they have nothing to give. And then we sit down with them for an hour and they have a blast.
So I,
So
I would love to tug on that thread just a little bit more because that is something that is, I feel like, really limiting in the world, which is there is a perception of what a scientist is, and it's a very one-dimensional view. And it's, they're in their lab coat, they're really stuffy, they're dry people. All they do is science all day long. And I think that's actually discourages a lot of people from getting into science because they feel like they are these complex multidimensional people and they can't be both that and a scientist. It's an either or choice.
Sure. And I think there's this also layered perception of that you have to be the 0.1% of the smartest people that you've ever interacted with in your life. I occasionally go and speak at these rural high schools, right? And like try to encourage kids to do science, right? And I have kind of a character that I do, like, hey, I came from a way worse school than this. I didn't get great grades, and I can do this, right? Like you can do this too. Why aren't you thinking about it, right? And so, but they have this perception, and it's like, okay, okay, the valedictorian, she's gonna do it, right? Like maybe she can be a scientist, but actually, and that's it. That's the only person there that can do it. But also that they're boring. I think that's also very important.
and And I know I say that we don't really have a focus, but sometimes I will catch myself like it feels like a PSA to like young researchers because I do think the current generation is more aware of self-care, balance, that there's more diversity, that there's more ways to be a scientist. And sometimes like I feel that like I know we probably say it, we'll do like just the last one, like emphasizing that not moving for your job is fine. right? Like I almost feel like in some way our these guests allow us to say these other things that you don't hear about are fine. And the people do it and they are, even if they're not like successful, like you said, by whatever standard that's crazy, they're sciencing, right? They're having a life. And so I think, yeah, that I think that is an important thing, is not just to show people they can do it, but also if you're a scientist, is to show them all the ways that you can do it. Yeah.
I think that's such a huge message. I'll just briefly mention I had my lab does do a lot of work with trying to bring opportunities to underserved communities and underserved students. And it's really one of the privileges of having that platform, is being able to do that. And I had a student one summer who, again, first generation student, his parents never finished high school, et cetera. And he came into the lab. And at the end of the summer, he came up and said, I I really was nervous about this. I had, I sort of thought that what scientists was they were all these like brilliant Albert Einstein like people, and now I see that that's not true. And I was like, thank you.
Like, I'm not, yeah, which part's a compliment?
But it really did change his trajectory. Like all of a sudden, science was approachable to him, and scientists were approachable, and something he could picture himself being. It wasn't this, off on a tier sort of concept. And so, even though he phrased it exceedingly badly, the impact was awesome.
Yeah.
Great.
I'd love to change gears just a second to think about other, you, Ben Orsburn, in addition to the podcast, you do have other communications that you do through your blog, et cetera. And I'm just curious about how the combination of being more active in communication has changed how you think about your research and yourself as a PI and scientist.
Oh, the communications, I mean, honestly, so the blog was a complete surprise, right? When I left my academic postdoc to go to, an applied industry postdoc at the NCI, right? There was going to be no chance to publish, right? And so, I had started this thing in my first postdoc where I was like, okay, I'm going to read a paper every day because I can't compete with anybody here because I'm not as smart as they are, but I'm going to just acquire all this information. And then I was like, oh, well, I'm just put it online, right? And I just kept doing that, and people sometimes, later found that. And it's funny how often I've used it recently. I have, I have an entire paper that was inspired by a Twitter conversation back when Twitter was good, and I acknowledge, it was a discussion of some work that Sam Payne had done, and then and instead of what you normally do with Sam Payne stuff, you just copy it word for word and publish it somewhere else, right? But I was like, okay, well, I'm going to reproduce this study on a time of flight instrument, and it's really fun. And I acknowledge the proteomics Twitter's community, specifically people that contributed to the conversation. One was an undergrad that just like chipped in. And so and it really shaped that entire study. And so I really do use that stuff. And sometimes, I like to put a preprint up that I know isn't complete and then sit back and see how people respond to it for the second draft. Right. So I think we can leverage those things. I'm not sure that it's the most, always like always maybe the smartest thing to do from a perception from your own marketing perspective, like putting something out that is like, whoa, okay, that's kind of dumb, right? Or this could use some serious work. But at the end of the day, if your goal is just like doing better science and you've got really thick skin, I just use it, right? So
Right.
that's kind of a ramble.
No, that was fantastic. And this concept of what is the role of what is your job as a scientist? And I think that aspect of our job is actually to take feedback and to be better. And so that it's not about the checkbox of the publishing of the papers. It's about making the best science we possibly can. And sometimes that gets lost along the way. And it's easy to let your ego get in the way and be like, oh man, I just got trashed. But that is actually, that productive discourse, that productive, not cynical, not mean, but productive critical evaluation is actually essential to us doing the best science. And so it's great that you actually have created an extra vehicle for doing
That was a lot smarter than what I said. I like how you said it better.
that. It's yours. I give it to you. I'm curious also, just in terms of you've through your long experiences in the field. This is not me calling you old, but both of you, how have you seen? Um, what are the trends that you've observed in proteomics in particular? How it has evolved, either through conversations with your guests or just your own observations of the field.
I mean, I think Ben's more. I mean, I can speak to like the show, right? Or I mean, I feel, we were talking earlier about like our lineage or whatever, like pedigree. I don't feel like I wasn't in proteomics, like in proteomics until really 2010, 2012. And I think that specific moment, a lot of things happened. We talk about, MS Convert, ProteoWizard kind of bringing together file formats. At the same time, you had a lot of instrument advances. That was a big moment. You had open data, Pride really became like a big deal. Later, we had preprints. Like, so now is a very different time. If you want to listen to funny things, listen to episodes when we have like older people talk about the 90s and I get everything wrong. But about the last 10 years, I think I don't feel like we need to talk so much about instrumentation, and we can talk more about biology. And the questions you ask within the show, I think the things that really excite me are the spatial, like I still go back to it, but like combining spatial metabolomics with proteomics, like how do you find new pathways? We take all of our results and we rely on these really pretty janky databases to try to provide insight. And I think we're finally graduating to be able to ask these really cool questions. That's my vibe on the field, is maybe less talk of numbers and more talk of science.
Yeah. I think that, it's been really cool to watch the field as it goes, right? And we're seeing more biological studies, we see a lot, we're of course still seeing I can get 8,000 proteins, I can get 8,015 proteins or whatever, right? But the fact that we're seeing a lot of biology happening, I think there are some things that the field has always done that haven't changed though. And we are the worst communicators in all of science, right? Like it's inventing acronyms, and it's like the you can hide as the mass spec wizard in the basement, right? And you can make a career being cantankerous and dismissive because people walk into your dungeon and they don't know all the acronyms, right? But when you see the people that are being really, really successful, they're ditching the acronyms. They're the better communicators because that cool biology is out there, and those people know all, like they have impactful questions. And if you can bridge that gap, right? And you see, Neil Kelleher has this whole TED Talk thing, right? And he never shows them. He never mentions a mass spectrum. He never shows one. Like there's nothing, right? It never comes up. And it's just like, and I just always think about that, like, how can we communicate better? Because I think we're still really, really bad at it.
Yeah.
But
So go ahead, Ben.
Well, I was just gonna say, I mean, you see that with, our favorite guests that, took us 70 episodes to realize are the people that don't do proteomics, because they'll show up and talk about these questions they're asking. And sometimes they'll say, God, wouldn't it be great if, I could measure these protein isoforms and compare it to my genomic data. I'm like, you can do that. You could do that 10 years ago. But again, and that's, Ben's then like, well, the reason is because we're not communicating, right? Like they're not even asking for, fancy things. I'm not, the spatial metabolomics, all that. They're
Yeah.
just asking for proteomics, and they don't think it's possible, maybe because something bad happened in 2008 when they were watching a talk and they just dismissed it for the next decade. But yeah, it's so talking to these non-experts is really fun because I think we see a lot of opportunities there. Um,
That's really interesting. Why do you think in particular that there are these communication barriers and both within the field and then beyond the field from proteomics to other? Is it that what we're doing is so hard and complicated? Is it
Yeah.
why is that barrier there?
It's oh, soapbox. I don't remember how to put it, but
Go for it.
we don't have a formalized education program in proteomics, right? Now, you can go to just about any university in the United States and get a genetics degree, right? You cannot get a proteomics degree anywhere. So we don't have this unified language. We don't have we don't have textbooks. We don't have this common language within ourselves, right? And so then you have these, where did where did you do your graduate work? Where did you do your postdoc, right? And the technologies that were developed there under that name versus something extremely similar that that was
Yes.
developed somewhere else. And I think we've got this, right? Like, so you've got this confusing nomenclature because we don't have this shared thing. And I think that's a lot of the problem,
Mm.
right? That just doesn't exist. So that's, yeah, I think that's,
I
Yeah.
Yeah, and I don't think I'm one like I think people have probably thought about it more like how did we end up where we are? But it's also just that, like even if you go back and you hear the stories, everyone was like they weren't a chemist, they weren't a biologist, they were this in between. So until very recently was this even like in a like kind of what Ben's saying, like it was a accepted field. It was almost like they were always in this weird no man's land. And then on top of that, just how hard it is, right? Like to do start to finish proteomics, it's like I got to be great in the wet side. I have to be basically an electrical engineer, physicist, whatever to run the instrument. Oh, and plumber. And it's just breaking. And
Yeah.
then to analyze the data, I'm probably some sort of bioinformatician programmer. And you've got people that are like siloed all over the world that are all being all these parts at once. And then someone comes in and is like, hey, can I run a sample? And we're always like, ugh. And to me, and again, I'm not one to speak to this, but I think that's the problem. It's like that entry point. Like people talk about how they want proteomics to do everything, but if someone runs and walks into your lab and says, I need you to do the sample, it is going to be like an hour conversation, and you're probably going to say no. If they walk into a genomics lab and say, run the sample, I think they're just going to say yes. And
Mm.
maybe this is me oversimplifying it, but that's for me is we are conditioned. Again, maybe it's all these exceptions and acronyms and difficulties, but there's so many ways to do it. And I think it just creates this barrier. If you talk about can universal methods or universal acquisition, people in our field, I don't think want that or like that.
Mm.
And it's like just I don't think it's not accomplishing maybe what you want it to accomplish. So
Yeah. No, that makes a lot of sense. And I think part of what you're describing is that there is this incredible fragmentation around how proteomics is done, just from lab to lab to lab to lab, because there are so many knobs and levers on the instrument and on the HPLC up front, and there are even more knobs and levers on how you do the sample prep, and so and everybody has experience from, oh, my lab does it this way, and my progeny do it this way, as you mentioned, and so it's very hard to say, okay, this is the way that proteomics should be done for asking this set of questions. Whereas I do think in genomics, there's a pretty standard QIAGEN kit you buy and you do a DNA extraction, and there's a pretty standard Illumina library prep kit. And so we just don't have that level of consistency. And maybe part of it is because we're trying to eke out. We just did a study in my lab just for reference where we were looking at different Eppendorfs or microfuge tubes from different vendors, and we're like, okay, which one of these gives us the least losses to?
oh, cool.
Um, and the difference was like 3% across many of the low bind tubes. It was not a large effect, but it was an effect. And I could imagine that, that might push us to say, oh, well, I could get 3% better performance if I use this thing at the cost of uniformity across the field.
Yeah.
Absolutely. Yeah.
Well, and that's I've heard it like the positive way of describing what we're talking about is we are in a innovating field. And like that's the thing. Like we're innovators. And that's great. But again, if your goal is to be like mainstream, at some point you gotta be like, listen, guys, like the term just turned 30 years old. Like maybe we can kind of pull it back a little bit. Just let it grow. Let it be old. It's 30. Like
Yeah.
it's getting towards middle age. Stop.
That's a really interesting point. So would you, I'm just going to ask you bluntly, would you declare the beginning of proteomics to be 1989 electrospray, or would you say it's the LCQ that came out, or are you 2D gels dating back even?
No, I mean go back to like the 1962 paper. This is that was Pauling and Zuckerkandl. Oh God, he's gonna kill me. But they were running tryptych digest on paper like TLC, whatever it was, and they were identifying these peptides. Proteins, the study of proteins always was the spotlight. Yeah, talk about 70s, 2D gels, later that became obviously bigger. It was bigger before it somehow just died, and it was genomics, right, in the 90s. So no, I mean, I think the study of proteins, yes, the term is Mark Wilkins, 1994, but I would go way back. We've always studied proteins and even proteins at scale. So it's always been important. I just don't know why no one, like, why are we not a topic? Like, that's my biggest pet peeve. Like, would you have like name, like what's your field? And there's like genomics, transcriptomics, everything, geology. There's never proteomics. There's never proteomics. Like, what is up, guys?
Okay, I have a very different take on this question. I would just, and the students I had at Hopkins were pharmacology students. They were using proteomics as a tool. They were getting pharmacology degrees, and I just like with very few exceptions, I would tell them not to read anything before 2017. And you can start with Dorda Becker-Jensen et al., like 12,000 proteins quantified in within the same amount of time that it took to do an Illumina high-seq run, right? So theoretical complete protein, because proteome, that ohm is, implies all the things, right? And I could do a crappy microarray in 2009 that would measure 23,000 transcripts, right? And it wasn't until 2017 that we saw that you didn't have to spend a month, right, to get to 12,000 proteins to get a peptide for every open reading frame. And so from then, I think that's when proteomics becomes a thing. It's a cool word, and I've often described it as it was kind of a lie. But it is
aspirational.
Yeah. So it was, there's protein mass spectrometry, right? And then we're at this point now where, like, proteomics is this thing where people are completing, like, you're not getting 50 or 100 at that kind of depth today, but we're getting close, right? We're seeing 8,000 proteins off some of these instruments that are doing, 100 speed. Right. So we're seeing, these other technologies are getting close to all the proteins that are there, and they're doing it realistically in a realistic time frame. And I think that's proteomics. So I think that's a relatively new thing.
That's a great take. I love that take.
And Magnus hates when I say this.
Well, I feel like I had some really lovely papers before that, and so I hate to discard all of those, including like my proteotypic peptide paper about what peptides you can actually see. But
it's easy for me because I didn't publish anything until recently.
But so I'd love to take this maybe one step further. So we talked about the barrier with regards to the scientific community, how we have genetics departments, but we don't have proteomics departments. which is a fascinating thought. I've always felt that where we are now is actually biochemistry hitting back at genetics, and so that proteomics sort of lived under the broader umbrella of biochemistry, and so that was good enough. But maybe that's not good enough. Maybe we need to go even harder and differentiate the general umbrella of biochemistry from protein sciences. You can come up with a better name for the department.
Hmm. I mean, I tell people I'm an analytical biochemist
Hmm.
because it feels more appropriate. And maybe that's still kind of aspirational, but like it feels more that's what I'm doing because proteomics is just, yeah, it's kind of nebulous.
Mhm.
And I don't know. I feel like, I mean, I tell this to strangers. I told it to a stranger this morning. They're like, what are you doing? I'm like, I'm an analytical biochemist. I measure your proteins in your blood.
Hmm.
That works.
Yeah.
So,
And so that concept of stranger. So we talked about communication within the scientific community, but what about the outside world? I, I'm curious about what are the barriers that you see, or what do you see with regards to proteomics in the outside world? Uh, is it there at all? If not, why not?
you know, well, here, I mean, you know, do you know how they re-released like they updated the Magic School Bus
I did
and
not know that, but now I'm
oh
excited.
it's really good. It's what's her face
McKinnon.
Kate,
Yeah.
yeah, and she's Mrs. Frizzle. But like in one of them they're like CRISPR editing DNA. And again, this show is for very small children, and my children totally they're fine with it. They're like, yeah, genes, messing with them gives you traits, boom. And I'm like, why can't we just talk about some proteins? Right? Like, how did that become a protein? No, it's there, you never hit that level. And I think too part of it, and I was kind of thinking about this, like even when I was talking to the stranger today, is um, I can sequence your genome today or tomorrow or last year. And that's just what it is, right? And that's kind of one of the reasons it's simpler, right? There's no caveat versus if I said I want to use your blood sample to understand something about you, it's like, well, is it 2 hours postprandial? Did you just have an infection? Like what time of the month is this? What season is this? What's your sex, age, blah, blah, blah. And it's just inherently this like fuzzy signal. And I think that's the problem too, is like it's hard to communicate that you say like I can do all this stuff and then they turn around and say like, okay, do it. And you're like, oh, well, hold up. I don't like normal. Like what are normal levels? That's well, what is I don't know what normal is. Like because I
That's
um
right. Well, because I think part of what you're talking about is that I can measure your genome and it's your genome.
yeah.
I measure it once and I'm good. But your proteome is different in every cell in your body at every moment of every day. And it's just a scale of complexity that is so much larger than the genome.
And Miss Frizzle is not going to be able to do that. Like it's too much, right? She's not going to be able to do it.
That's
I
so
don't
so
know.
my hot take as to why and I'm curious to get your reaction to it, is that we had movies like
Oh, Jurassic
Gattaca
Park.
and Jurassic
Oh,
Park, and
Yeah,
they
yeah.
it's
were
Jurassic
all
Park.
all about the genome. And
Yeah.
so it's out there, and we have not had a movie about the proteome.
Mm. There was
Sure
there was The
haven't.
Expanse. There was that spoiler. There was that episode of The Expanse where they
Mhm.
put the sample into the thing and it spits out uniprot numbers, and that's the answer. And like two people caught that.
Ooh.
Just what to say. No one cares. But okay, keep going. Yeah, we need it. Keep okay, Osborne, go.
Yeah, no, I was so encouraged when there was the big article in Forbes, right? And that's 2020, around 2019, something like that, right? That the that it would be cool if they'd had, they hadn't had a handmade mass spectrometer from the 70s, as this like really terrifying looking thing that's obviously, hand built, as the picture. But, but yeah, it did attract interest, right? I think the title was the massive proteome or the massive investment opportunity of proteomics or something like that, right? And so you do see these things sneaking in. And I think, as much as I'm concerned about these big targeted panels, right? Thermo just signed a thing, right? Where Olink just set up a 660,000 sample, right, screen. It's going to take absolutely forever at the current throughput, but that's putting proteomics in the public consciousness, right? We're thinking about the proteins there. If people keep repeating that, 90% of drug targets are targeting proteins, right? If those things, I think that's going to get that to kind of the public consciousness, but it is slow and it's complicated and kind of boring, right?
Well, and it's kind of too like we already measure proteins, right? Like what's your A1C number, right? Like the protein. Like we're already doing it. But yeah, I think there's a disconnect there because like you
Yeah.
said, like we're all about Jurassic Park, I think was huge. Like they
Well,
saw the A, T, C's, and G's. Like we need the equivalent of that.
Well, so let's
That's
let's just dive into this a little bit more because it's clearly not enough, right? What happened with we're going to go deep into Jurassic Park lore now.
ooh,
So what happened is they actually saw not a genetic mutation. They saw probably an epigenetic change that led to changes in dinosaur behavior, which were almost certainly be caused by changes in their proteome. And so really Jurassic Park was lying. It was not genetic alterations that were driving dinosaurs going nuts. It was proteomic alterations.
missed opportunity.
I haven't seen it since the 90s, but that makes a lot of sense, right? um, huh.
Well, and that's right because and we found I mean yeah, so we can use like the reptile example, right? Like they could be male or female based on temperature or that's like how I think alligators do that, right? Like and I think in Jurassic Park it was I don't even know why they
Like
were
frogs.
trying
They were like crossed with frogs in some way. And yes.
but yeah, like so there are these and again this came up on the show like someone had this problem where it was this very evident this is why you study proteins, right? Like your DNA is fine. And then you had to know this, you had to be able to study the trafficking or turnover. Was it the ApoE thing? But yeah, like it's not just do you have it? Because this is the one that gets me is like we've got these SNPs and we're like, oh well, that's SNP and now you're toast. I'm like, is it? Like because it's really complicated and it does all this other stuff. And yeah, I feel and yeah, we do complex pop sci. We know people could understand it. I don't know how you convince them, like, not only is it important, I mean, take the COVID vaccine. It made your body make a spike protein. I think everyone had this weird lesson in how that works. I don't think it helped.
Yes.
Like, I don't think anyone still understands
Yeah.
like what a protein is. They're like, yeah, tracker,
Well,
I don't
I mean,
know.
in part because your cereal box does list protein, right? Your yogurt says this is how
Mm,
much protein. And so I
Oh,
wonder if
yeah.
that's actually working against us. It does not say how much DNA is in your yogurt. There's probably some there, I'd guess.
Yeah. I
Uh,
bet.
some
Yeah.
some left over. And so I think that just even the concept that there are 20,000 plus different proteins, if you were
Sure.
to ask people in society, most people wouldn't know that.
Yeah.
yeah.
No question.
And I get the dietary protein thing a lot. Like if you start talking about proteins, like, oh, you mean like plant protein or animal protein? Like, what is that? I mean, again, even think about like you can go and buy collagen, or you
Mhm.
can buy like it's all around us. Yeah, just
I can tell by your hair that you are an avid user of collagen.
all the
Um,
time.
yeah.
Just today, just did it. Um,
So I think that's a question. So as a community, are we doing enough to actually to influence the broader public conversation and bring proteomics to the forefront?
Okay. I think Ben and I are kind of maxed out,
Okay.
but as a community, I think we could do more.
no, you mean like we need to do more? But like think
Yeah.
about like if you had a proteomics department, if you had a national institute, what's the NHLG, RGI or whatever, like if you had the
Yes.
proteomics version of that, I think that's when you start getting they start like you said, it's like exposure. Like, we know where we send things, get it sequenced at the sequencing core, and then people are like, how does my doctor know, oh, I need to send this to the protein sequencing core.
Right.
I think it just needs to, if it would help though if you had named resources around, and then it would start sinking into people. But man, I don't know. I mean, my kids know proteins, but like, yeah, get Miss Frizzle to do it.
Yeah.
Wait, no, wait. I mean, we're not answering your question. I mean, okay, Osborne, your dream is we need the program, right? We need a natural degree, right?
Yeah.
If people could get genetics degrees
Mhm.
forever,
Yeah.
right? So, is it a degree? Do they see it out and
Yep.
about, funding institutes, whatever?
Mhm. Right. Are there news articles saying about how a there are articles all the time about, oh, researchers found the gene that controls baldness. Um, but you just don't see the same scale of, hey, we found the protein that is responsible for this disease.
Yeah.
Or
No.
But that's why all these, the last however many months for the UK Biobank is such a big deal. Like, and maybe I think you were getting at that is like, this is how you get mainstream is we did samples on 53,000 people and now it's hitting the news. Like, but then you run that risk, which is probably something that happened in the early 2000s, is you need it not to fail, right? You need it to, okay, so it's making an entrance again.
yes.
How do we make it not like be bad?
Yes. Well,
Yeah.
I think maybe as a community, we are also a little too careful there. We had a really bad false start. But how many thousands of papers about RNA-seq have necessarily led to incredible scientific discoveries?
Yeah. No, people are quick to brush the RNA microarray under the table, and there were hundreds of millions of dollars spent on RNA microarrays that really weren't all that good at the time, right? The machining tolerance wasn't good enough. The algorithms weren't in place to do it really well. And not a lot of stuff came from those, right? You can do 100x sequence coverage, right? Like pretty easily now on these instruments, right? So, but we were able to do 20x sequence coverage 15 years ago.
Mm-hmm.
Is doing 100x solving the problem, or is it just filling up hard drives, right? But we're a little more forgiving of the fact that hasn't had a lot of success, and then proteomics is only just now starting to have like some like really tangible effects, right? That are getting out there.
Though there will be breakthroughs. Like I do, if we were going to say from a technological standpoint, starting 2017, wherever you want to start, 2015, it's been really good for us. Now we're hitting the point where it's going to be really good for society, right? There's going to be drugs. I feel like the GLP-1 thing, like we can argue like it's a protein, like it's going to help enter the public conscious. We're going to have these big studies happen that are going to enter, companies are going to start offering these assays that are not just like a thing. I mean, so it feels like it should be entering public conscience as a real thing, like as just a day-to-day concept. So
That's a perfect place to end. I look forward to the conversation when somebody walks up to me and instead of saying, "Oh yeah, that's in your DNA, that's in your genes," they're like, "Oh, that's in your amino acids, that's in your proteins."
like your PTM's
cool.
wacky.
Yeah.
So thank you both so much for joining me today. This was super fun. I really appreciated all of your perspectives and so thank you again.
Thanks for having us
Thank
on.
you. This is lovely.
Absolutely. And as a reminder to the audience, this has been Translating Proteomics. Please join the conversation, hit us up with comments, send us email, and let's keep the conversation going.
We hope you enjoyed the Translating Proteomics podcast, brought to you by Nautilus Biotechnology. To contact us or for further information, please email translatingproteomics@nautilus.bio or scan the QR code on your screen.